Hello, this is part 2 of our presentation for Lab 2 measurements. To begin where we left off, lastly I showed you myself adding samples to the electrophoresis tray. Now this is a video of Regine adding samples to the electrophoresis tray. Again, I apologize for my slow computer. Alright, I hope we did fairly well. Moving on. Okay, so to backtrack a little bit, before we add the samples to the gel, we let the gel solidify. And after the gel solidified, we added the 1 times TAE solution to the tray, almost filling it, not letting it overflow like another group in our class. But yeah, and then after that we added the dyes, and then we added a current to it. Of course, we know that there's one positive electrode and one negative electrode. So we powered those, and after a couple minutes, we started seeing short migrations, but this was only a few minutes after, so this is what happened later. After 19 minutes of allowing them to run, this was our final result in the gel. As you can see, a couple moved all the way to the edge, which was impressive, which is what we wanted, and the others moved a significant amount. So this is our table showing the final results. The dye xylene cyanol moved in the positive direction. It moved 24 millimeters, and if you do the calculations, because it was 19 minutes, that would be about 1.26 millimeters per minute. Bromophenol blue also moved in the positive direction for about 34 millimeters, and that comes out to be 1.79 millimeters per minute. Orange G also moved in the positive direction for 40 millimeters, which calculates to 2.11 millimeters per minute. Safranin O was the only one that moved in the negative direction, and it moved in that direction for 30 millimeters, which comes out to 1.58 millimeters per minute. And then the mix of the 4 dyes moved in the positive direction, which makes sense because the majority were moving in the positive direction. That migrated 32 millimeters, and the rate of migration was 1.68 millimeters per minute. So in conclusion, the dye molecule with the greatest weight is xylene cyanol because it moved the slowest at 1.26 millimeters per minute. The dye molecule with the lowest weight is orange G because it moved the fastest at 2.11 millimeters per minute. And all the molecules except for safranin O were negatively charged because they moved towards the positive electrode, because of course opposites attract. Safranin O moved towards the negative electrode, and that makes sense because that means safranin O would be positive because it moved towards the negative electrode. So this means that our hypothesis would explain the movement of the molecules, because we said that larger molecules would move slower and smaller molecules would move faster, which is apparently what happened. And positive molecules move towards the negative electrode, and negative molecules move towards the positive electrode. Okay, so that is the end of our presentation.